Hello, my name is Leonard.
And I'm Camille Wetmore.
And we did our enzyme project on sucrase-isomaltase. Sucrase-isomaltase is a glucose-saccharase enzyme composed of two subunits of sucrase and isomaltase. It breaks down carbohydrates and carboxylic activity that play vital roles in the catalytic mechanisms in the sucrase-isomaltase complex, also known as the Si complex. They're both found in the active site of the enzyme complex, and the conduritol B epoxide is the actual active site for the inhibitors of the sucrase-isomaltase enzyme. So the origin of the Si complex is in your small intestines and the epithelial cells, and the way that it is transported through the cell is that it comes in through one side of the transmembrane and is directed directly into the ribosome endoplasmic reticulum, so the rough ER. It is checked there for any toxins. It's kind of its way through the beginning part of the cell, and it goes directly to the Golgi apparatus. That is where it's packaged and sent off to the basal lamina side of the cell and is then excreted from the cell from that membrane.
So the function of sucrase-isomaltase is it catalyzes the final step of the carbohydrate digestion by breaking down disaccharides and oligosaccharides into simple monosaccharides or simple sugars so that the body can further absorb it and be able to use it as energy. So here you can see sucrose. So sucrose is made up of glucose and fructose, and to bring them together is by taking out water, and to separate them you're adding in water. So hydrolysis and condensation reactions. Okay, so the mechanism of sucrase-isomaltase. So as stated before, glucose and fructose are started bonding together, and when they come together with the enzyme sucrase-isomaltase, you add in water, which breaks the bond apart so that there's two products that exit out of the enzyme substrate complex. And so then fructose and glucose can go into the body and be able to be used as energy, and then the enzyme can then be able to be reused so that the cycle can continue onward.
So as we talked about earlier, the whole transport of the Si complex through the epithelial cells, that is very vital to its actual function. We came across a primary source where it talks about the congenital sucrase-isomaltase deficiency, also known as CSID, as you hear us refer to it as later on in the presentation. CSID is a very rare genetic disorder that someone has when in their intestines they don't have the catalytic activity of the Si complex, and it doesn't break down the glucose and the fructose as it should, so it doesn't give you that energy from the carbohydrates as you normally would if you had the Si complex in your small intestines. It affects the ability to actually digest the sugars and starches, therefore giving you the energy that you need from those carbohydrates. It's sucrase subunit dependent, and what that means is because you have a two subunit enzyme, the sucrase and the isomaltase, it actually just directly affects the sucrase subunit of the enzyme. So the isomaltase doesn't have the deficiency associated with it. And there are 5 different phenotypes of CSID, and what that means is that your 5 different phenotypes give you different outward appearances. So you can have stomach cramps or diarrhea, abdominal pain. Any of those are different phenotypes of CSID itself. And it's the transport competent molecule. So when you're talking about the whole transport through the epithelial cell itself, that is when it is missorted and mistransported throughout the cell that actually gives you this CSID deficiency. And we also will talk later about the single point mutation in the sucrase subunit itself, where they found that CSID is also relevant. That's where you get it. There's a different mutation in its DNA where it should not have it. So the methods of this paper, they used patients who had a long life history of abdominal pain, abdominal cramping, diarrhea, all of those different phenotypes that you have when you have CSID. Those were people who were then assessed to be a part of this trial. Or this research. So what they did to assess them was they used the breath hydrogen test. And you can see that here in this picture. So what a breath hydrogen test is that these patients were forced to eat different foods that had a high concentration of fructose or glucose. And normally when your fructose or your glucose is digested in your small intestines, your SI complex then breaks them down as they normally should, and you breathe out a normal amount of methane, hydrogen, and carbon dioxide. Well, when you have CSID, you have a buildup of bacteria in your small intestines where your SI complex is not functioning the way that it should be. So when you do breathe through the breathalyzer test for your hydrogen, methane, and your CO2, your levels are often elevated due to the bacteria in your intestines. And you can see that here on the graph. People with CSID have a higher breathalyzing test of your methane, your hydrogen, and your CO2. They also used a sucrose tolerance test, and so people who had gone through and were tested positive through the breath hydrogen test, they will also then use a sucrose tolerance test to make sure that they had an intolerance for sucrose in their intestines. And the way that they did that, they took biopsies that they later used on in the research. They used biopsies to make sure that they were sucrose deficient. And like I said, 5 different biopsies were taken of each person's intestinal cells, and one of them was put through an immunoelectron microscopy. That wasn't really a lot of how they found the CSID, so that wasn't a lot of the paper. What they actually focused on was the frozen liquid nitrogen that they used for RNA replication, and we'll talk a little bit more about that later on, but what that does is that it gives you a platform to have a single-stranded DNA to find that single point mutation that we touched on earlier. The other 3 biopsies were biosynthetically labeled and put into organ tissue culture dishes, and they used that to run an SDS page, which we talked a little bit about in class, and that is how they figured out the mistransport of the SI complex through epithelial cells.
Okay, so for the results, so they used the SDS page to measure the effects of the anti-SI antibodies, which is endo-H and endo-F. And so at 210, kilodaltons, thank you. At 210, you can see that it is detected in the endoplasmic reticulum. So you can see in the control, they're all seen in the 210. And then at 245, that's when it's detected in the Golgi body of the cell. And so you can see the control, they're all active at 245. But with the CSIDs, they are not, they sort of come to the 210, but they don't come at all to the Golgi body. So as Laura said, there has to be some sort of miscommunications within the cell, and that the communication within the cell or the cell mechanism is just off, and so that it's just not being able to be detected in those areas. So then also the RNA isolation, which is done by liquid nitrogen, it helps isolate where the point mutation is. So they use a single strand DNA, and then they find the point mutation, which was where glutamine was replaced by proline. And so it was an AC conversion at nucleotide 3298. And so it found that while the encoding of the sucrase portion of the SI complex.
So the conclusion of all of these results in CSID itself, there are several different phenotypes, as we mentioned. They were all analyzed, so all the different phenotypes, people who had different symptoms of CSID, were all tested in the same environments. And all cases show that the transport incompetence, missorting, and the enzyme malfunction to the bilateral membrane are all misinterpreted. And we could see that, as Camille was talking about earlier with the SDS page, those 2 control groups were actually other enzymes who have similar cell activity as the SI complex. So as those being the control, they were able to rule out any generic cellular misfunction that the SI could have brought along with it. So comparing it to 2 other enzymes who have very similar cellular activity, ruled out the cellular activity transport itself being the main cause of CSID, showing that other enzymes don't have that same enzymatic activity deficiency. It also characterizes the point mutation in the single-stranded DNA from the liquid nitrogen isolation of the RNA of the Si complex. And like Camille touched on before, it's only a part of the sucrase subunit of the Si complex. So when they ran the Endo-H and the Endo-F, they ran both the sucrase and the isomaltase subunits and only found the deficiency to be relevant in the sucrase subunit. When run against the isomaltase subunit, no enzymatic activity was found, therefore they found the deficiency in the sucrase. And this Q1098P, that is the amino acid is 1098P. That's the amino acid that is switched from the glutamine to the proline that gives it its genetic mutation, where people who have CSID have the proline amino acid at 1098, and people who lack the CSID deficiency, they have a glutamine as it should be in the single-stranded DNA. As you can see here, this is a representation of the variable. It's a variable point mutation, and you can see that it's the glutamine to the proline, which means that you have variable phenotypes. So if you have a point mutation and it's there's no effect, or it has usually very serious, very specific phenotypes, whereas CSID is a very variable phenotypic deficiency of sucrase-isomaltase. And if you have any more questions, or you want to look at the paper that we looked at, if you have any more questions about the methods or anything like that, please check out our paper. It's very interesting, and we really appreciate you watching our video. Thank you.
Thank you.